or in this unit, we have 3 different chapters. Chapter on water, chapter on carbon, and we have the chapter on macromolecules. So I think the first chapter for us to start with is the chapter on carbon. You guys can find some place to sit. Just go sit down somewhere. All right?
Are you going to be making a recording of this?
I am recording this.
So you want to do one of them?
No.
Oh, you're recording?
Yes.
Yeah.
There you go. All right. So first thing, carbon. The most important term for you to link with carbon is going to be tetravalence. Right? For carbon to have tetravalence, what that means is that carbon contains 4 valence electrons. Right? And since carbon has 4 valence electrons, it is able to bond with 4 other atoms. Right? That is what is so special about carbon. Right? That is how the structure of carbon leads to its unique characteristics, which lead to diversity in life. Right? It's all about carbon's tetravalence. It's all about carbon being able to bond with 4 other atoms. So what is the role of carbon in biological or in organic molecules? Right? Carbon is going to form the backbone. Right? It's going to form the core central structure that is going to give every organic molecule its shape. Any molecule that contains carbon, we refer to it as an organic molecule. Right? Organic meaning carbon containing. When we look at these organic molecules, there are 2 factors that are going to determine the properties of an organic molecule. One is going to be the carbon backbone, right? Specifically, the arrangement of carbons in the backbone is going to determine the shape of the molecule, and the shape determines its function or its properties, what it's able to do. We can see molecules with all different shapes, right? You could have linear molecules, you could have branching molecules, you can have carbon rings, you could have double bonds, you could have triple bonds, you can have all different arrangement of carbons in the backbone. The second factor that's going to lead to the properties of an organic molecule are going to be the functional groups. The functional groups are the groups that branch off of the carbon backbone, right? Like we said in class, you don't need to memorize the functional groups, but you should be able to recognize them in specific molecules. The functional groups are important because they are the part of the organic molecule that's going to participate in chemical reactions. Right, so it's the functional groups that react. So again, there are 2 factors that determine the properties of an organic molecule. One is the arrangement of carbons in the backbone. 2 is going to be the specific functional groups that it contains. And that's because those functional groups are the groups that react with other molecules. If we want to look at a study of the relationship between structure and function, we can look at isomers. Right? Isomers are going to be molecules that have the same molecular formula, but they have a different arrangement. So they have the exact same atoms and they have the exact same number of atoms. But somehow those atoms are arranged differently. Therefore, they have a different shape. And by having a different arrangement of atoms, they are therefore going to have different properties. There are some really good examples of isomers that we see often. A good example that you should know is glucose and fructose. They both have the same molecular formula. They're both C6H12O6. However, the way that they are arranged is different. If we look at glucose, it looks like a hexagon. Where if we look at fructose, it looks like a pentagon. So even though they have the same atoms, they have the same number of atoms, they have the same molecular formula, because their arrangement of atoms is different, they're going to have very, very different properties. All right? We talked about 3 different types of isomers. We talked about structural isomers, geometric isomers, and enantiomers. Structural isomers have a different arrangement of carbons in the backbone. We call them structural isomers. The second type of isomer is a geometric isomer. Geometric isomers have the same arrangement of carbons in the backbone, but they have a different arrangement of functional groups. And finally, the last type of isomer is going to be an enantiomer. Enantiomers are sometimes called stereoisomers, and they are mirror images, or they are left and right hands. So there are slight differences between each type of isomer. Every kind of isomer, you have the same molecular formula. That is always true. They just have slight differences in their arrangement. In a structural isomer, the carbon backbone is arranged differently. In a geometric isomer, the functional groups are arranged differently. Enantiomers look identical superficially, but when you look at them up close, they are mirror images, and they therefore have different properties. Any questions on carbon? Go ahead.
Can you go over the difference between structural and geometric?
Yeah. A structural isomer has the same molecular formula, but a different arrangement of carbons in the backbone.
Okay.
All right? So glucose and fructose are structural isomers because their carbon backbone making up the ring is arranged differently. Geometric isomers, again, you'd have the same carbon backbone, but the groups that branch off of them are different. And if you remember with geometric isomers, you might have 2 carbons double bonded together, and sometimes you would have the functional groups on the same side, and sometimes you'd have the functional groups on opposite sides, right? If they're on the same side, what type of formation do we call that? We call that cis. If they're on opposite sides, what do we call that? Trans. And we learned how important that was when we looked at trans fats. Right? When you look at a trans fat and you look at the fatty acid chains, the functional groups are on opposite sides of that double bond. And that's what ends up making the fatty acid very linear and solid at room temperature. Right? That's what makes trans fats so bad for you. I know I'm moving fast. I'm just trying to get everything in. Are we okay?
Yes.
This would be an example of geometric isomers. same carbon backbone, but a different arrangement of the functional groups. All right. Macromolecules. So we'll go through each type. First type of macromolecule we talked about is the carbohydrate. First of all, carbs are only made up of 3 atoms, right? They're made up of carbon, hydrogen, and oxygen. The monomer of a carbohydrate is a monosaccharide. Monosaccharides are things like glucose is a monosaccharide. Fructose is a monosaccharide. Ribose is a monosaccharide. You can always tell if something is a carbohydrate because carbohydrates are always going to have a 1 to 2 to 1 arrangement. Whether it's C5H10O5 or C6H12O6, you always have a 1 to 2 to 1 arrangement. So if I give you a data table and it's got a bunch of atoms and you see that 1 to 2 to 1 arrangement, right? You can bet that it's a carbohydrate. Did we all hear that? You might see something like that tomorrow. When you put 2 monosaccharides together, you make a disaccharide. And the most typical disaccharide you're going to see is going to be sucrose, table sugar. When you put 2 monosaccharides together, what chemical reaction do you have to perform? Dehydration synthesis. Right? So remember, dehydration synthesis is the removal of what? Water. Right? So you're removing water, and whenever you perform dehydration synthesis and you remove water, you are creating a bond. All right, we will see this over and over and over and over again in this course. Right? Anytime that you are making a bond, you're always making it through dehydration, the removal of water. Anytime you break a bond, period, you're always breaking the bond through the same reaction. What's it called? Hydrolysis. Right? Hydro meaning water, lyse meaning to split. Right? You add water in to break a chemical bond, right? This is happening over and over again. Anytime you're breaking a bond, hydrolysis. Anytime you're making a bond, dehydration. What's going to change from macromolecule to macromolecule is the name of the bond, right? So in carbohydrates, we refer to it as a glycosidic bond, right? So glycosidic bonds are made between monosaccharides, and glycosidic bonds are made through dehydration synthesis. We're doing okay? All right. You put many monosaccharides together, you make a polysaccharide. There are many different polysaccharides that we talked about. You have starch. You have glycogen. Starch and glycogen are basically identical to each other. They're both used to store energy. But plants store energy as starch. Animals store energy as glycogen. They are both polysaccharides. They are both made up of very long chains of glucose. Does anybody remember the name of the type of glycosidic bond that is found actually in both starch and glycogen?
Alpha.
It's an alpha glycosidic bond. Good. And we are able to break down alpha glycosidic bonds. So we're able to break down starch. We're able to break down glycogen. And they're able to give us long, sustained energy. The other polysaccharide that we saw is cellulose. Right? Cellulose is what we commonly refer to as fiber in our diet. And cellulose is going to contain beta glycosidic bonds. And we are not able to break down beta glycosidic bonds. So when we ingest cellulose or when we ingest fiber, it's going to go through our digestive system unbroken down, undigested, because we cannot break those beta bonds. You guys could come in and stay by the door. All right. The last polysaccharide that we talked about is called chitin. And chitin is kind of a modified polysaccharide. It's structural, right? It's found in fungi. It's also found in insects, right? They use chitin as a structural component, right? Plants use cellulose as a structural component. I'm trying to think if there's anything else you need to know about carbs. That's it. We good? Lipids. Let's move on. You guys can go sit at a lab table if you want. All right, lipids. Lipids are also made up of C, H, and O. So they're made up of the same atoms that carbs are. How can you tell a difference between a carb and a lipid? Lipids have a very high ratio of carbon and hydrogen. They have a lot of C and H, and they have very little O. Are lipids polar or nonpolar? They're nonpolar. So are they hydrophilic or hydrophobic? They're hydrophobic. How about carbohydrates? Are carbohydrates polar or nonpolar? Polar. They're polar. So are carbs hydrophilic or hydrophobic? They're philic, right? They dissolve in water. So lipids have a very high quantity of carbon and hydrogen and a very low quantity of oxygen. And lipids are used primarily for energy storage. The polymer of a lipid is called a triglyceride. And a triglyceride is made up of 3 fatty acids attached to a glycerol. The fatty acids are attached to the glycerol through dehydration synthesis. And what is the name of the bond found inside of a triglyceride? It's called ester bond. Good. So you have ester bonds found within a triglyceride. When we look at fatty acids, they fit into 2 different categories. You can have a saturated fatty acid, or you could have an unsaturated fat. Saturated fats are going to have all single bonds. By having all single bonds, they are going to have the maximum amount of hydrogens. Right? So they are saturated with hydrogen as a result of having all single bonds. The single bonds make very linear chains, And as a result, saturated fats are going to be solid at room temperature. So if you look at animal fat, like the fat on a steak or butter, that is made up of saturated fat, all single bonds, maximum hydrogen, linear chains. Unsaturated fats are created through a double bond. So when you create a double bond, what do you have to lose to have that double bond? Hydrogen. Right? So unsaturated fats have less hydrogen, and that double bond is going to create a bend in the chain. And when you bend the chain, it's going to make it less dense, and therefore make it what at room temperature? Liquid. All right, so unsaturated fats are created through a double bond. The double bond creates a bend, and that bend is what makes an unsaturated fat liquid at room temperature, where the saturated fats are solid at room temperature. All right, we talked about some other specific types of lipids. We talked about phospholipids. Phospholipids are going to compose the cell membrane. When we look at a phospholipid, it is going to have a polar head and two nonpolar tails. So that polar head is made up of a phosphate group, PO4, and the two nonpolar tails are made up of fatty acids. So a phospholipid is a really unique molecule because it is half polar, half nonpolar. Or you could also say it is half hydrophilic and half hydrophobic. One of the things that we talked about with phospholipids is that they contain carbon, hydrogen, oxygen, and phospholipids are also going to contain phosphorus. We also talked about cholesterol. Cholesterol is also a non-polar lipid. We said that cholesterol is found within your cell membranes. Cholesterol is also used to make steroid hormones like testosterone, estrogen, and progesterone. Too much cholesterol in your bloodstream can lead to the formation of plaques. We call it atherosclerosis, right? Plaque formation due to buildup of cholesterol. But cholesterol is another type of lipid. Moving on. Next, we have proteins, right? The most important of the macromolecules. All right, we spent a lot of time talking about the role of proteins in the body. Proteins do everything, right? Your carbs and your lipids, they're all about energy. Proteins do all the rest. Proteins are going to have carbon, hydrogen, oxygen. They're going to have nitrogen, and they're also going to have sulfur. Sulfur is a defining characteristic of a protein. The monomer of a protein is called an amino acid. Most amino acids are all the same. They all have a central carbon. They have a carboxyl group. They have an amino group. They have a hydrogen. And the fourth group is the R group. And it's that R group that makes one amino acid different from another. There are 20 different amino acids, and they differ in their R group. The R group can make it polar, can make it non-polar, can make it acidic, can make it basic, can make it all different kinds of stuff. If you want to put two amino acids together, you have the carboxyl group of one amino acid react with the amino group of another amino acid. You have an H and an OH, and together you have the removal of water. So what reaction are we performing right here? Dehydration synthesis. You have the removal of water, and you're forming chemical bond, right? And the name of the bond between amino acids is the peptide bond. Now, it is really, 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 really important for tomorrow that you understand the levels of structure within a protein. So if we look at the 4 levels of protein structure, you begin with its primary structure, which is its most simple structure, its most basic structure, right? Its most fundamental structure. Its primary structure is the linear sequence of amino acids, right? And remember, ultimately, that is what is going to make one protein different from another. Different proteins have different sequences of amino acids. That is fundamentally what makes them different. So our primary structure is just the linear sequence of amino acids peptide bonded together. Now these amino acids begin to interact with each other. And depending upon their sequence, they're going to start to form hydrogen bonds. And these hydrogen bonds can cause alpha helixes to form. It could form beta pleated sheets, right? So the hydrogen bonding is going to cause the sequence of amino acids to be bent and twisted in different ways. So the secondary structure is going to be due to the formation of hydrogen bonds, right? Creating either alpha helixes or beta pleated sheets. Now, the amino acids are going to continue to interact. And you're going to get all kinds of bonds. You might have alpha helixes here, beta pleated sheets here, alpha helixes there, beta pleated sheets here, right? And this creates the tertiary structure. And this tertiary structure is created by all different types of bonds, right? We could see ionic bonds. We could see more hydrogen bonds. We could see hydrophobic interactions. That's when two nonpolar parts are attracted to each other. We see disulfide bridges. Right? We have all these different types of bonds that are being created. And it creates this super highly folded, very specific shape. And this thing that we see right here is what we call a polypeptide subunit. And that polypeptide subunit is not a protein. To get to the protein, we get to the 4th level of structure, which is the quaternary structure. You're going to put multiple polypeptides together. And once you have multiple polypeptides together, now you have your finalized protein. Some proteins have 2 polypeptide subunits. We call them dimers. Some, like collagen, have 3. Some, like hemoglobin, have 4. Different proteins have differing numbers of polypeptide subunits. But ultimately, the most important level of structure is going to be your primary structure. It's all about the sequence of amino acids, right? Because the sequence of amino acids ultimately is going to determine the shape of your protein, and the shape of your protein is going to determine what it's able to do. It's going to determine that protein's properties. Really, really important, right? We saw a good example of that, right, with sickle cell anemia. Sickle cell anemia is caused by a change in just how many amino acids? Just one, right? One out of hundreds of amino acids. Just by changing one amino acid in the primary structure, that causes a change in the secondary structure, which causes a change in the tertiary structure, which then causes a change in the quaternary structure. And that's going to change the protein as a whole, right? In this case, leading to sickle cell anemia. There are two environmental factors that are going to affect the functioning of this protein. One is going to be temperature, and the other is going to be pH. Now, we're just introducing them now. We'll talk about them more when we talk about enzymes. But whenever you change the temperature that a protein is in, or you change the pH that a protein is in, you run the risk of denaturing the protein. Right? So proteins get denatured. That means that all those chemical bonds that are holding it together are broken, and that protein unravels. And when a protein unravels, it's functionally dead. It will no longer do what it's supposed to do. So all proteins work at very specific temperatures, very specific pHs. A good example of denaturing a protein is cooking it. I always think of cooking an egg. Right? Eggs contain a protein called albumin. It's clear. And when you cook it, you fry an egg, it turns white. It turns white as a result of those proteins denaturing. You might see that question on the test tomorrow. I think that's everything of proteins. All right, the last macromolecule, which we barely covered, just a small little intro, is going to be the nucleic acid. All right. Nucleic acids are going to be DNA and RNA. Nucleic acids have carbon, hydrogen, oxygen, nitrogen, and phosphorus. So that's how you can tell the difference between a nucleic acid and a protein. Nucleic acids have phosphorus, proteins have sulfur. The monomer of a nucleic acid is going to be a nucleotide. When we look at a nucleotide, it's going to be made up of a phosphate, a sugar, and a nitrogenous base. Right? They compose nucleotides. And we'll see them a lot more in our genetics unit. All right. That is all of carbon. That is all of macromolecules.